In the early 1920s, deep in the sun-bleached quarries of Taung, South Africa, a discovery was waiting beneath the layers of ancient limestone, a discovery that would shake the foundations of science. Let's dig into it. Workers chiseled through rock, unaware that inside one of the blocks lay a face frozen in time. When the crate arrived at the University of Witwatersrand, anatomist Raymond Dart opened it with curiosity of a man who sensed opportunity. What he found was astonishing. The tiny skull of a child, its features a mosaic of familiar and alien. The brain was small, no larger than that of a chimpanzee, yet the position of the foramen magnum, the hole where the spine meets the skull, told a different story. This child had walked upright. Dart held in his hands a being that defied the scientific dogma of the age. He named it Australopithecus africanus, the southern ape of Africa, and dared to propose that humanity's earliest roots were African, not European. But the world wasn't ready. The Piltdown Man hoax still dominated the narrative, a supposed early human from England with a large brain and primitive jaw, perfectly fitting the Eurocentric belief that intelligence came first. Dart's fossil contradicted everything. It was African, it was small-brained, and it was upright. The scientific establishment dismissed him. Journals refused his papers. Scholars mocked his conclusions. For decades, Dart stood alone, defending a fossil that the world refused to accept. But time has a way of revealing truth. As more fossils emerged from caves in South Africa, and as the Piltdown Man was exposed as a fraud, the Taung child rose from obscurity to become one of the most important discoveries in the history of paleoanthropology. It was the first whispers of a story that would eventually lead us to a story not written in Europe, but in Africa. The Taung child represents one of the most significant turning points in the study of human evolution. Dated to approximately 2.8 million years ago, the specimen preserves both a facial skeleton and an endocast, a natural mold of the brain's surface. This endocast provides a rare insight into early hominin neuroanatomy. The brain volume, estimated at 405 cc's, is within the range of modern chimpanzees, yet the morphology reveals early signs of human-like reorganization. The lunate sulcus, a major brain fold associated with visual processing, appears shifted posteriorly, suggesting an expansion of parietal association areas, regions linked to spatial reasoning and early cognitive complexity. The position of the foramen magnum is one of the strongest indicators of habitual bipedalism. In quadrupedal apes, it sits towards the back of the skull. In the Taung child, it's positioned centrally beneath the cranium, aligning the head atop an upright spine. This single anatomical feature was Dart's most compelling argument for bipedal locomotion. Dart's interpretation was initially rejected due to the dominance of the Piltdown fossil, which falsely suggested brain expansion preceded bipedalism. The Taung child contradicted this sequence, showing that upright walking evolved millions of years before significant brain enlargement. It wasn't until the 1940s and 1950s when Robert Broom uncovered an adult Australopithecus africanus specimen at Sterkfontein and Makapansgat that Dart's claim gained traction. These confirmed that the Taung child was not an anomaly, but part of a broader species with consistent anatomical traits. Taphonomic analysis later revealed that the Taung child was likely killed by a large raptor. Puncture marks in the orbits match those produced by modern crowned eagles, providing rare evidence of predation on early hominins. This insight reshaped our understanding of the ecological pressures faced by Australopiths. The Taung child remains a cornerstone of paleoanthropology, the first fossil to demonstrate that early hominins were small-brained bipeds, and the first to firmly place human origins in Africa. South Africa's Cradle of Humankind is a landscape carved by time, a rolling expanse of dolomite hills, sun-baked grasslands, and hidden caverns that plunge deep into the earth. To the untrained eye, it looks peaceful, almost ordinary. But beneath the soil lies one of the richest archives of ancient life on this planet. Millions of years ago, these caves were natural traps. Rainwater dissolved the limestone, forming vertical shafts that opened like mouths in the ground. Animals that wandered too close slipped and fell into the darkness. Predators dragged their kills inside to feed. Over time, bones, sediment, and debris accumulated layer upon layer, sealing the past in stone. It was in these caves, Sterkfontein, Makapansgat, Gladys Vale, and others, that the story of Australopithecus africanus slowly emerged. Sterkfontein, with its labyrinth of chambers, yielded hundreds of hominin fossils, including the iconic Miss Pless, whose face became the symbol of early African ancestry. Makapansgat offered its own mysteries. Bone fragments once thought to be used as tools, a strange pebble shaped like a human face, carried miles from its origin. Gladys Vale, rediscovered after decades of silence, revealed new remains that expanded the species' known range. These caves are more than fossil sites. They're time capsules. Inside them, the air feels heavy, as if the past still lingers in the shadows. Every bone, every tooth, every fragment of ancient life tells a story of survival, death, and the relentless march of evolution. The cradle of humankind is not just a place where fossils are found. It is a place where the boundary between the past and the present feels thin, where echoes of ancient footsteps seem to whisper through the stone. The fossil sites associated with Australopithecus africanus are among the most scientifically important paleoanthropological localities in the world. These sites are primarily located within the dolomitic limestone formations of South Africa's Mulmani subgroup, where karst processes created extensive cave systems. Over millions of years, these caves accumulated breccia, a cemented mixture of sediment, bones, and rock fragments that preserved hominin remains in exceptional detail. Sterkfontein is the most prolific source of Australopithecus africanus fossils. The site contains multiple stratigraphic members, with member 4 being the richest in Australopith remains. Radiometric dating places member 4 between 2.6 and 2.1 million years old, though some estimates extend slightly earlier. The assemblage includes cranial, dental, and postcranial elements, providing a comprehensive view of the species' anatomy. The STS-5 cranium, Miss Pless, and the STS-14 postcranial skeleton are the most significant finds, offering critical evidence for bipedal locomotion and cranial morphology. Makapansgat Limeworks, located in the Limpopo province, preserves a different ecological context. The site's member 3 deposits contain numerous Australopithecus africanus fossils, along with a diverse faunal assemblage. Early interpretations by Raymond Dart suggested that bone accumulations represented deliberate tool use. That so-called osteodontochoratic culture. Later taphonomic studies disprove this, showing that carnivores were responsible for the bone concentration. However, the site remains crucial for understanding Australopithecology and predator-prey dynamics. The Makapansgat pebble, A jasperite stone resembling a human face was found in the same deposits. Geographical analysis shows it originated several kilometers away, suggesting it was transported, possibly by Australopithecus africanus. While not evidence of symbolic behavior, this raises intriguing questions about early pattern recognition. Gladys Vale Cave, rediscovered in the 1990s, produced the first new hominin fossils from the cradle of humankind in nearly half a century. The remains include fragments attributed to Australopithecus africanus, expanding the known geographic distribution of the species. The site also preserves faunal and environmental data that help reconstruct the paleoecology of the region. The cave sites share common taphonomic processes. Vertical shafts acted as natural traps. Carnivores contributed to bone accumulations. Sediments gradually encased remains in breccia. Calcite flowstones sealed deposits, aiding preservation. These processes created a unique fossil record that captures not only hominid anatomy, but also broader ecological context. Predators, prey species, vegetation indicators, and climate signatures. These sites form a multi-layer archive of early hominin life, offering unparalleled insight into the biology, behavior, and environment of Australopithecus africanus. To understand Australopithecus africanus, we must reconstruct the body that carried them through the world of predators, shifting climates, and evolutionary crossroads. Their bones tell a story of transition, a species no longer fully ape, yet not fully human. When we look at their skulls, we see the echoes of our distant past, a small brain case, rounded and compact, holding a mind just beginning to recognize itself. The face projects forward, but not as dramatically as in earlier Australopiths. The cheekbones flare gently, the jaws robust yet refined, and the teeth, thick enameled and human-shaped, speak of a diet that demanded strength and versatility. But it's the body that reveals the most profound transformation. Their pelvis is short and wide, reshaped by millions of years of evolutionary pressure to support upright walking. The femur angles inward, bringing the knees beneath the center of gravity, allowing them to stride across the savanna with surprising efficiency. The spine curves in a gentle S shape, balancing the torso above the hips and cushioning each step. These are hallmarks of a committed biped, a creature that walked upright as naturally as we do. Yet their arms tell a different story. Long, powerful, and built for climbing. They reveal a species still deeply connected to the trees. Their fingers are curved, their shoulders high and mobile, their upper body strong enough to pull themselves into branches when danger approached. They lived in two worlds at once, the open ground and the forest canopy, navigating both with a grace that modern humans have long since lost. Their feet, too, reflect this duality. The heel is broad and human-like, built to absorb the impact of walking. But the midfoot remains flexible, capable of gripping branches. They were walkers and climbers, ground dwellers and tree dwellers, a species perfectly adapted to a landscape that demanded versatility. In Australopithecus africanus, we see the blueprint of humanity emerging. Not in a sudden leap, but in a gradual mosaic transformation. Their bodies were experiments in evolution, testing new forms, new functions, and new possibilities. They were the bridge between what we were and what we would become. The anatomy of Australopithecus africanus provides one of the clearest windows into the evolutionary transition from arboreal primates to fully terrestrial bipeds. Their cranial, dental, and postcranial morphology reflect a complex interplay of ancestral traits and derived adaptations. The cranial capacity of Australopithecus africanus ranges from 420 to 510 cc's, overlapping with Australopithecus afarensis, but showing a subtle increase in cortical reorganization. Endocasts revealed posterior displacement of the lunate sulcus, expansion of parietal association areas, early development of Broca's cap region, and increased hemispheric asymmetry. These features suggest enhanced visuospatial integration, improved motor planning, and early foundations for proto-linguistic capabilities. The cranial vault is more rounded than earlier Australopiths, with reduced nuchal musculature indicating a more balanced head posture consistent with habitual bipedalism. The face of Australopithecus africanus is less prognathic than that of Afarensis, with the reduced subnasal projection, moderate zygomatic flare, parabolic dental arcade, thick enamel, and reduced canine dimorphism. Microwear patterns show a mixed diet of soft fruits, hard objects, and fibrous vegetation. Enamel isotopes indicate consumption of both C3 and C4 plants, reflecting ecological flexibility. The pelvis of Australopithecus africanus is one of the most important indicators of bipedalism. The ilia are short and broad, reoriented laterally to stabilize the trunk during single-leg stance. The sacrum is wide, supporting lumbar lordosis. The femoral neck is elongated and the valgus angle is pronounced, aligning with the knees beneath the body's center of mass. The vertical column shows lumbar wedging consistent with an S-shaped curvature, essential for shock absorption and upright posture. Despite their bipedal adaptations, Australopithecus africanus retains numerous arboreal traits. Long humeri, cranially oriented glenoid fossa, robust clavicles, and curved manual phalanges. These features indicate powerful climbing abilities, suggesting that arboreal refuge remained a key survival strategy. The hand exhibits a well-developed oppens pollicis, enhanced precision grip capabilities, and intermediate phalangeal curvature. The foot combines a human-like calcaneus, partially adducted halix, and a flexible midfoot. This mosaic of structure supports both terrestrial bipedalism and arboreal grasping. The males average 40 to 50 kilograms, while females average 30 to 40, with height estimates of 110 to 140 centimeters. Dimorphism levels suggest moderate male competition, possibly with multi-male, multi-female social groups. The anatomy of Australopithecus africanus reflects a species adapted to efficient terrestrial bipedalism, arboreal climbing for safety and foraging, dietary flexibility, and complex social behavior. This combination of traits positions Australopithecus africanus as a key transitional form in hominid evolution, bridging the gap between earlier Australopiths and the emergence of genus Homo. To understand Australopithecus africanus, we must step into the world they inhabited, a world of shifting landscapes, unpredictable seasons, and constant danger. 3 million years ago, South Africa was not the dry, open savanna we imagine it today. It was a mosaic of environments, patches of dense woodland clustered around rivers, open grasslands stretching towards the horizon, rocky outcrops rising from the earth, and seasonal wetlands that came alive with life after the rains. This was a land in transition, shaped by cycles of drought and abundance, where survival demanded flexibility. In this ever-changing world, Australopithecus africanus became the master of adaptation. Their teeth tell the story, thick enamel built to withstand the grinding of tough vegetation, molars shaped for crushing, and wear patterns that reveal a diet far more varied than once believed. They ate fruits when the trees were heavy with ripening sweetness. They dug roots and tubers when the dry season stripped the land bare. They cracked nuts and seeds when other foods grew scarce. They may have even scavenged meat when opportunity allowed, using stones or bones to break open marrow-rich bones left behind by predators. But food wasn't the only part of their struggle. The land was alive with predators. Saber-toothed cats crouched in the tall grass. Hyenas prowled the night,
and leopards stalked the rocky cliffs. Even the skies were dangerous. The Taung child with its haunting puncture marks remind us that the giant eagles hunted early hominins, swooping down with silent wings to snatch the vulnerable. For Australopithecus africanus, every day was a negotiation between hunger and safety. They moved through this landscape with a dual strategy, walking upright across the ground, scanning for danger, and climbing trees when threats drew near. Their bodies were built for both worlds. Feet strong enough for long-distance walking, hands curved for gripping branches. They lived in small groups, relying on one another for protection, foraging, and the shared knowledge of where food and water could be found. In this world of uncertainty, Australopithecus africanus survived not through strength alone, but through adaptability. They were opportunists, improvisers, and survivors. Creatures shaped by a land that demanded resilience. Their ability to thrive in such a dynamic environment laid the groundwork for flexibility that would one day define the human lineage. The diet and ecology of Australopithecus africanus has been reconstructed through a combination of microwear analysis, stable isotope studies, dental morphology, faunal associations, and paleoenvironmental reconstruction. Together, these data reveal a species with remarkable dietary flexibility and ecological adaptability. Microscopic wear patterns on molars show a combination of fine scratches associated with soft fruits and leaves, deep pits associated with hard object feeding, and complex textures indicating fallback foods during seasonal scarcity. This suggests a diet that varied seasonally and regionally, consistent with the generalist foraging strategy. Carbon isotope ratios extracted from the tooth enamel reveal a mixed diet incorporating both C3 and C4 plants, C3 being fruits, leaves, woodland vegetation, and C4 being grasses, sedges, and open habitat resources. This isotope signature distinguishes Australopithecus africanus from more specialized hominids like Paleoanthropus robustus, which show stronger C4 signals. The mixed isotope profile indicates Australopithecus africanus exploited multiple ecological niches and was not restricted to forested environments. The thick enamel and robust molars of Australopithecus africanus are adaptations for processing mechanically challenging foods. Finite element analysis of molar structure suggests that the teeth were capable of withstanding high bite forces, supporting the hypothesis that hard objects, nuts, seeds, and possibly underground storage organs, were important fallback foods. Faunal assemblages from Sterkfontein and Makapansgat indicate heterogeneous environment with woodland and forest patches, open grasslands, rocky escarpments, and seasonal wetlands. The presence of both arboreal and terrestrial fauna support the interpretation of a mixed habitat. Pollen and phytolith analysis further confirm the coexistence of grasses and woody vegetation. Predators contemporary with Africanus included Dinofelis, saber-toothed cats, Megantarian, early Panthera species, Pachycrocuta, giant hyenas, and large raptors. Taphonomic evidence, including tooth marks and puncture patterns, indicate that predation pressure was significant. Juveniles were particularly vulnerable, as demonstrated by the Taung child's cranial damage consistent with eagle predation. Postcranial anatomy suggests dual locomotor repertoire, habitual terrestrial bipedalism, and retained arboreal competence. Combination allowed Africanus to exploit both ground and canopy resources, enhancing survival in predator-rich environments. Based on primate analogs and anatomical evidence, Africanus likely engaged in opportunistic scavenging, digging for tubers, fruit harvesting, nut cracking, and even termite probing. These behaviors reflect a flexible foraging strategy that would later characterize early Homo. The adaptability of Australopithecus africanus to diverse habitats and fluctuating climates underscores the importance as a transitional species. Its ecological versatility foreshadows the behavioral flexibility that would become the hallmark of the human lineage. To imagine the social world of Australopithecus africanus is to step into the realm where survival depended not only on strength or speed, but on connection. These early hominins did not walk the landscape alone. They moved in small, tightly knit groups, families, bound by necessity, instinct, and the beginnings of something deeper: cooperation. In the shifting mosaic of woodlands and grasslands, every member mattered. Alone, Australopith was vulnerable. But a group could watch the horizon together, share food, protect the young, and navigate the dangers of a world filled with predators. Their social lives were likely rich with communication. Though they lacked language as we know it, their voices would have filled the air with calls, grunts, and rhythmic vocalizations, sounds that conveyed alarm, reassurance, curiosity, and intent. Their faces, more expressive than those of modern apes, could communicate subtle emotions: fear, excitement, affection. Their hands, capable of delicate precision, may have even played a role in gestural communication, pointing, reaching, or signaling to others in a way that bridged the gap between instinct and intention. With their groups, relationships were everything. Mothers carried their infants for years, nurturing them through the long childhood that demanded patience and teaching. Youngsters learned by watching how to climb, how to forage, how to recognize danger, how to read the moods of others. Adults formed alliances, cooperated in foraging, and shared knowledge of the landscape. Males may have competed for status, but cooperation likely outweighed aggression. The pressures of predation and environmental unpredictability favored groups that worked together, not those torn apart by conflict. Grooming, a behavior shared by many primates, would have been central to their social world. Fingers combing through fur, removing debris, soothing tensions. These simple acts strengthen bonds and reinforce trust. In moments of rest, they likely huddled together for warmth and safety, their bodies forming a living circle against the night. Their social structure was not yet human, but it was no longer purely ape. It was something between, a foundation upon which the complexities of human society may one day be built. In their interactions, we glimpse the earliest roots of empathy, cooperation, and shared identity. They were learning slowly and subtly that survival was not an individual struggle, but a collective one. Reconstructing the social behavior of Australopithecus africanus requires synthesizing evidence from anatomy, comparative primatology, paleoecology, and developmental biology. While direct behavior evidence is absent, multiple lines of inference allow for a scientifically grounded reconstruction of their social world. Based on body size dimorphism, predation pressure, and ecological modeling, Australopithecus africanus likely lived in a multi-male, multi-female groups of between 10 and 30. This structure parallels that of chimpanzees and early human foragers, where group cohesion provides protection and enhances resource acquisition. Moderate sexual dimorphism in body mass and canine size suggests some degree of male-to-male competition, but not the extreme polygyny seen in gorillas. Likely a flexible mating system, possibly involving pair bonds or cooperative breeding. This intermediate pattern aligns with the species that balance competition with cooperation. Dental eruption patterns and cranial growth rates indicate a prolonged juvenile period, relative to apes. Extended childhood implies increased parental investment, opportunities for social learning, and reliance on group support for offspring survival. This developmental trajectory is a precursor to the extended childhood seen in Homo. The social complexity inferred for Australopithecus africanus represents a critical stage in hominid evolution. Their combination of extended childhood, cooperative behavior, enhanced communication, and group cohesion laid the groundwork for the emergence of highly social, culturally rich societies of Homo.
To understand the significance of Australopithecus africanus, we must follow them as they take one of the most important steps in the history of life on Earth. The one step onto two legs. Bipedalism was not simply a way of moving. It was a revolution written into bone and muscle. A transformation that reshaped the body from the ground up. When Australopithecus africanus walked across the ancient South African landscape, they carried with them the blueprint of a future that would one day define our species. Imagine them moving through the tall grasses, their heads raising above the vegetation as they scanned for predators. Their gait was steady, rhythmic, and efficient, each step conserving energy in a world where survival often depended on endurance. Their pelvis was short and bowl-shaped, cradling their internal organs as they walked upright. Their spine curved gently, absorbing the shock of each footfall. Their legs swung beneath them in a smooth arc, powered by muscles arranged in a pattern that no ape possesses. Yet, even as they walked like humans, they climbed like apes. Their arms remained long and strong, their shoulders high and mobile, fingers curved for gripping branches. When danger approached, a leopard stalking from the shadows, a saber-toothed cat crouched in the grass, they could retreat into the trees with the agility of their ancestors. They lived in two worlds at once, navigating the ground with confidence and the canopy with ease. Bipedalism opened new possibilities. It changed the way they saw the world. Some potential drivers of bipedal evolution include thermoregulation, or reduced solar exposure, efficient foraging and carrying, predator detection, long-distance travel, and freeing the hands for tool use. While no hypothesis fully explains the transition, Africanus represents a mature stage in evolution of bipedalism, integrating multiple selective pressures into a coherent anatomical package. Compared to Afarensis, Africanus shows more derived pelvic morphology, improved hip abductor mechanics, enhanced lumbar curvature. These differences suggest a refinement of bipedal locomotion over time. Bipedal adaptations of Africanus laid the biometrical foundation for the emergence of Homo, whose increased brain size and tool use would depend on energetic efficiency and manual freedom provided by upright walking. Thank you for watching the first part in what may be a 4-part series on Australopithecus africanus. When all the videos are done, I will combine them and make one long documentary-style movie, as well as having the parts available for watching separately. Depending on the amount of information for the next set of fossils, the video format may fall into this style as well. Once again, thanks for watching. Don't forget to like, comment, and subscribe, and stay tuned for more deep dives here on Fossil Matrix.